do not want to keep my ink. All right. So the next one we're talking about today, which is perfect because of lab this week. We are going to be going through our urinary system. Okay, cool. We might get into endocrine today. This is awesome. So, urinary system. This one is short and sweet because there's not much I can talk to you guys about in the urinary system while we're in anatomy. The urinary system is super important in like just staying alive because of what it does. Okay, when we think of the urinary system, typically the main organ we think of is our kidney. And the kidney has some incredible roles outside of just what we think of, which is excretion, right? Making urine. The process of urine formation is actually quite complicated, as you will learn in physiology. We'll talk for one slide about it today. But really what's going on is we are filtering blood, and we're taking things like waste and toxins and excess fluid, and we're putting it into the urine. And then we're absorbing things that we want to keep in our body. And what those things are changes depending on what's going on in your body at any given time. The one thing we overlook when it comes to the kidney is its regulatory function. So it does it two ways. Because the kidney is really involved in managing the composition of your blood, as it's pulling and pushing things out of the kidney and into the urine, or out of the urine and into the bloodstream, it changes the ions that are in your blood. It changes the pH of your blood. It changes the volume of your blood. So your kidneys are involved in, things like your blood pressure. In fact, when you learn your little blood pressure cascade in physiology, your kidneys are involved with that, with some important hormones and chemicals. So it can help regulate blood pressure. It helps maintain your acid-base balance. And all these things are super critical to maintaining homeostasis or staying healthy. The other things that our kidneys do is they release two hormones. So we've already talked about erythropoietin. Remember, this is red blood cell formation. Remember that root word here, erythro, or eryth, is red, like erythema, redness of the skin. The other one is calcitriol. This is the active form of vitamin D. Remember this guy when we get to the endocrine system. He will come up again, okay? So it's the active form of vitamin D. Now what we're going to do in this unit, we're not going to be learning about urine formation. That's too much. But we're going to be going through all the major structures of the kidney, the vasculature of the kidney, okay? And we'll be talking a lot about the actual unit of the kidney that's doing all of this filtration called the nephron. And then we'll spend a little bit of time looking at our ureters, our bladder, and the urethra, and then of course a little bit about the development, okay? As usual, I will post our objectives for this particular chapter online when I post the video later tonight, okay? Excuse me. So let's get into it. First things first, what does a kidney look like? Where is a kidney found? So your kidneys do not lie in the peritoneal cavity. They are actually what we call retroperitoneal, which means they lie behind it, okay? So they are actually outside that peritoneal cavity. They are retroperitoneal. They live in the retroperitoneum. They're found just below near your rib cage, okay? They're kind of at odd angles with one another. They're not super symmetrical. And they are connected and hang out in place through multiple different layers of like fascia and fat that kind of keeps them where they're supposed to be. The outermost layer of a kidney, so if we took off all that fascia and connective tissue and fat, we are left with this brown outside called the capsule. Okay, this is what's connecting the kidney to its sort of connections with the connective tissue and fascia, and it helps protect the kidney as well. This region here, let me switch my colors. This region here, where we see vessels and tubes coming in and coming out, that's called the hilum. Remember, we've seen a hilum with the trachea, which is where it branched. And we saw the hilum of the lung, which is very similar to this, where we had the bronchi and we had our vasculature and stuff coming in and out of the lung and that medial aspect. So the hilum is just how stuff gets in and out. Okay, the ureter and the renal artery and vein. Now, when you do a kidney dissection, you basically make a frontal section. So when you guys watch that kidney dissection video, they do a frontal section and open the kidney up like a little book, and it looks like this. Kidneys have regions and they have structures. So when you're taking quizzes, when you're taking the exam, you want to make sure to read the question. Am I asking for a structure or am I asking for a region? So the outermost region of the kidney is called the cortex. So the cortex is the outermost layer. It's that light color that you see here. Okay. Deep to that, you're going to have the renal medulla. Okay. This is the middle area of the kidney. And then the innermost region, just before we hit the ureter, is the renal pelvis. So we have our pelvis, the medulla, and the cortex. And this connection or orientation, I should say, of cortex medulla is very, very common throughout the human body. Cortex on the outside, medulla on the inside. Now, the columns that I have highlighted here, columns are simply where we have cortex in between these pyramids. So here's one pyramid. They're really easy to remember because they look like pyramids, okay? And then the cortex that you would see in between, that's a renal column, okay? And lobes, because as with everything, we have to subdivide because it's not anatomy if we don't. If we took one pyramid and we took half the cortex on one side and half the cortex on the other side, this is a lobe. That will be very important when we get to vasculature. So the lobes are basically like half the cortex on one side of a pyramid and half the cortex on the other side. We also have some more structures, because why not? When we look at our pyramid here, you'll notice that we have one area that's really pointy, this area here. That's called the renal papilla. Remember, a papilla or papilla is a finger-like projection. What's it projecting into? Well, it's projecting into these guys here. So right here and right here, we have these little cups. They kind of look like this. This is called a minor calyx. A little cup, okay? And these minor calyces merge into one another to form major calyces. So it's like you were trying to fill up a small glass, maybe of orange juice or something, and realize that, oh wow, I have more orange juice than I have cup space. You are going to take that and put it into a larger cup. So what these calyces are doing is they're not catching orange juice, they're catching urine that's being made by the unit that we call the nephron. So let me do a little bit of erasure here. The nephron is what we call the functional unit of the kidney. That's what this guy is right here. A little bit bigger than I usually can see. What we mean by functional unit is this is the component of the organ that's doing the organ's job. And in this case, changing blood composition, creating urine, things like that. The end tube here, and I'll talk to you about it a little bit more and we can zoom in again. You'll see there's a little yellow tube here in the renal papilla. That's the papillary duct. That's where it gathers all the urine from that pyramid and drains it into the calyx. So one of the things I really like about this diagram is it shows you the pathway. So we go collecting duct, papillary duct, minor, major calyx, renal pelvis, ureter, urinary bladder. So it shows you the pathway that urine takes as it goes out of the kidney. Okay? Now, one of the things about the kidney is it's filtering blood. And it's getting a ton of blood. About 20% of your cardiac output goes to your kidney, which means your kidneys are going to be very vascular organs. You thought you were done. Just a few more blood vessels. Kidney vasculature, as I've said in lab this week, is often daunting to students because, as you can see, there's a lot here, okay? But just like with the vasculature of our systemic vessels, they tend to tell you where they are, okay? So let's just run through what these mean. I have a mnemonic for you, and then I'll show you where they are in the kidney. So left and right renal artery, I'm not even gonna bother. We know where those are. Those are bringing the blood into the kidney. Segmental. This is where the renal artery segments, right? It fragments into smaller vessels. They're gonna head through the pelvis and they're gonna turn into interlobar arteries. Remember, inter means between, and lobar means lobe. So remember those lobes that we saw where we had the pyramid and the cortex on either side? The interlobar artery runs between those lobes. Arcuate, keyword here is arc. These arc, oops, not off. They arc over the pyramids. And then the last arteries we have are the cortical radiate. Cortical, cortex, the outer layer of the kidney, and radiate means it radiates, so it like permeates the whole cortex, okay? These guys are really teeny, 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 tiny, often look like speckles. And that brings us into the nephron. So the nephron, and once again, we'll be able to look at the nephron again, it has some vessels coming into it. Afferent coming in. This is often bigger. The glomerulus, as you'll see soon, is a ball of capillaries. This is where blood is filtered. And then efferent, you want to think exit. So this is going to exit the glomerulus. And this is often small. However, as you will learn in physiology, those shapes can change depending on what your body needs. Now, as we leave the glomerulus, so we've filtered our blood, there's still stuff in it, okay? We don't put all of it into the nephron. It's gonna exit and start our actual capillaries, which are the paratubular capillaries and vasa recta that we talked about in lab this week, or will, if I haven't seen you yet. And that brings us into our venous system. It's basically the exact same thing we saw with our arterial system. We just go backwards. Cortical radiate, arcuate, interlobar, renal vein, and then back to the inferior vena cava to the heart. The one thing that's missing is there is no segmental vein. The inner lobar runs all the way through the medulla, through the pelvis. There is no segmental vein. Okay, everything else is paired except the segmental vein and segmental artery, because there's no segmental vein. So let's go ahead and kind of put a picture to this, and then I'll give you guys my little mnemonic. Okay, so picture. Let's review them, and then we'll do the mnemonic. So here's the renal artery, and you can see how it segments. These are the segmental arteries. They're going to turn in to interlobar arteries as they go between the lobes. And so we go interlobar artery. We're going to arc up and over the pyramid as an arcuate artery, and then radiate into the cortex as a cortical radiate artery. The nephron vasculature, that's what's in this little dotted box. Take a look at that more when we look at the nephron next. And then we exit. We go through the cortical radiate veins, arcuate vein, interlobar vein, remember no segmental, and then the renal vein and the inferior vena cava. So how about a mnemonic? Randy swam in a cool and gorgeously illuminated pool containing an inflatable red iguana. If you remember that, you'll remember the order of operations. Randy swam in a cool and gorgeously illuminated pool containing an inflatable red iguana. Renal artery, segmental artery, interlobar artery, arcuate artery, cortical radiate, afferent arterial, glomerulus, efferent arterial, peritubular capillaries and vasa recta, cortical radiate vein, arcuate vein, interlobar vein, renal vein, inferior vena cava. So there is your little mnemonic for the kidney vasculature. And once again, remember, it tells you, they tell you what they look like, where they're going, or what's happening, okay? And this is good because we also have to know this in lab. So as you guys kind of work through the models, PAL and things like that, you'll be able to kind of get a better feeling for these, okay? All right, so now that we've kind of looked at the larger components of the kidney that we could see if we did a dissection, we're going to look at the nephron. Okay, remember the nephron is the functional unit. And what the nephron is a series of tubules. And actually, I'm going to draw this on the whiteboard just because it's, I'm going to draw a little different of a diagram that's a little bit easier to see. Okay, got that. So what I'm drawing is the exact same thing, basically, that's on your PowerPoint, but it's been sort of pulled out and straightened. So you can kind of initially learn the structure, okay? All right, so let's run through these. In the nephron, we have our vasculature, okay, right? Here's my afferent arterial. AA is arterial. This is my bundle of capillaries. We'll take a closer look at it soon. Okay. Does high blood pressure damage the glomerulus, the nephron, or both? It does technically damage both. You're totally right, Nicole. So with high blood pressure, what's happening is your capillaries can really only withstand a certain level of pressure. And as you have untreated high blood pressure, you're repeatedly putting high pressure blood into this really delicate capillary system, the glomerulus, and they can rupture. And so if I rupture the glomerulus, I have a really difficult time filtering my blood. And so it's going to diminish the ability for the kidneys to do their job. So that's why untreated high blood pressure can lead to kidney damage. Excellent question. So the blood's going to come in through the afferent arteriole, get filtered out through the glomerulus, and then whatever's left over exits the efferent. So where's the blood actually going? The blood is getting caught in this capsule that we call Bowman's cup or Bowman's capsule. And the glomerulus and Bowman's cup together is called the renal corpuscle. So the renal corpuscle houses the glomerulus and Bowman's capsule together. So we filter the blood, Bowman's cup catches it, and then it sends it down to our first part of the tubule called the proximal convoluted tubule. Proximal means close. In this case, it's not referring to a joint attachment. It's referring to the glomerulus. So this is the close. Convoluted, like if somebody has a really convoluted story, instead of getting from point A to point B, they kind of zigzag everywhere and you get lost in the story. So convoluted is twisty. So this is the close, twisty tubule. From here, we head down what's called the loop of Henle. So we have the descending limb, and we have the ascending limb. And yes, this is important because in physiology, and we'll see this in a minute when we look at the histology here, the descending limb and the ascending limbs of the loop of Henle have very different functions. So I just want to kind of get you guys prepared for that, to not think that they're the same thing. They have different roles, different things happen. So we go down the loop of Henle, we go up the loop of Henle, and then we hit our far twisty tubule, which is the distal convoluted tubule. Remember, distal means further away. So in this case, this is the tubule that is furthest from the glomerulus. Now, the reason I draw it this way is because you can visualize that. When you actually look at models or actual anatomical drawings, the nephron doesn't sit like this. The tubules all kind of sit on top of each other. So it kind of gets really hard to understand. So if we pull it out this way, we can get that the proximal convoluted tubule is the one that's attached to the renal corpuscle, and the distal convoluted tubule is the one that's attached to the collecting duct. And you'll notice I have a lot of offshoots here with the collecting duct. These would be connected to other nephrons. So the collecting duct is like I-5, big, long highway running through like the West Coast. And these would be like on-ramps that are going to be gathering urine from other nephrons. And the collecting ducts all come together and they'll dump at the very end into the papillary duct. Okay, so this is our drawing of a nephron. And once again, I'm going to save this and I will put this on our lecture webpage so you guys can have this to review or to copy down. Okay, so it will not be gone forever. So let's go back to the PowerPoint. And you can see what I mean by when we look at it anatomically, it's really hard to figure out who's who. So if you learn it with that simple drawing first, then you can start applying it to these more complex anatomical structures, okay? For our class, for our purposes, we don't really need to know what's really happening at each of these stages. I mean, besides that, the glomerulus is where we filter blood. All the other tubules are pulling and pushing things out depending on what the body needs. So we don't have to get as specific as we would in physiology. So let's look at the glomerular capsule. Remember the other name for this, oops, I don't have a pen. Other name for this is the brain dead, is the renal Bowman's cup. That's how I learned it, so that's the one I always remember. Bowman's cup or Bowman's capsule. 12 different ways to describe it. So when you look at the capsule, it's two-layered. It has a visceral layer and a parietal layer. And the visceral layer is the part that's literally sitting on top of the glomerulus. It has these cool cells called podocytes, foot cells. And the podocytes have these appendages that you can kind of see here, like this, that extend outward and wrap around the actual capillary. These are called pedicels, means foot-like projection. Remember, pedal means foot. So it's as if you took your hand, and if this is my podocyte, this would be like the cell body, and then all of my fingers are going to be the pedicels, and I can wrap it around my glomerulus, which is my sparkling water. Okay, so these would be pedicels. See how there's still some gaps between there? That will be important in a minute. And then we have an outer layer, which is going to be this part here. That's going to line sort of the cup that's catching everything. So the parietal layer lines the cup, and then the visceral layer is what's on top of the glomerulus. And it's important we understand that as the glomerulus comes in, it's not just capillaries like we've looked at before. They're very special capillaries, and they have to have these layers around them because the function, once again, of the glomerulus is filtration. So if we zoomed in on a cross section of our glomerulus, we would see not just the capillary wall, which is here, and the slits within the capillary wall. Remember, the type of capillary that we see here in the kidney is a fenestrated capillary. If you recall from chapter 14, if we were to look at one of those cells, it had all these holes in it. So if you can fit through the hole, you're getting out of the capillary. But we don't want everything to come out of our bloodstream. When we're doing glomerular filtration, even though it's size dependent, it's just if you fit, you can get through, we don't have things like blood cells going through. We don't want to see that. I don't need big proteins going through. The big proteins and things like that, and ions, they'll get pushed out later, but we never want red blood cells going through, right? Because then that means I'm losing my ability to carry oxygen. So they can't fit through here. Outside of that endothelium, we have the basement membrane, also known as the basal lamina. Okay, so that's going to be this layer here. That's between our capillary endothelium and those pedicels. It kind of acts like a barrier for ions, more or less, so they can't slip through all of them. And then you'll notice the holes now get smaller. So you might have been able to fit through here, but you'll get rejected once you hit the pedicels, okay? So once you've made it, if you can fit through both the fenestrations, you can get through the basal lamina, and you can get through the pedicels, you're now in Bowman's cup, and you're going to make your way through the nephron. But here's the thing. Things like water and glucose can fit through. But those are two very important compounds that we often do not want to just get rid of, okay? So they actually do get reabsorbed. So even though we're not really mitigating at the beginning who can come in, we will decide based off of our body's needs as you move through the nephron who needs to be put back into the bloodstream and stay in the body, and who can stay or get put into the nephron as we move through, okay? So here's a better drawing looking at the whole structure. We can see the Swiss cheese here, right? That's the endothelium of the glomerulus. We have the basement membrane here, or the basal lamina, same thing. And then we can see the podocyte and the little slits. So that's that filtration slit. That would have been the space between my fingers here. That would be the filtration slit. So if you can fit through all of this, you're going to be in the nephron. If you can't, you're going to go out the efferent arteriole, and then if we need to, we'll put you into the nephron. And if we don't need to, you're just going to go around the circulatory system. Okay? So that brings us to histology, everybody's favorite. Remember, histology is an important thing to understand as we look at organs because it's their most basic level of building. And the tissue lets us know the major function, more or less, of that particular organ or structure. This looks like a lot. It's going to be really easy to remember. Every portion of your renal tubule, the proximal convoluted tubule, the ascending limb of the loop of Henle, most of your distal convoluted tubule, and the collecting duct are all simple cuboidal, which remember during our first unit of lab and lecture, one of the places you would find simple cuboidal epithelium was lining the kidney tubules. The weird one is going to be, excuse me, the descending limb. The descending limb of the loop of Henle is made of simple squamous. That's because the descending limb and ascending limbs have different roles when it comes to who can move and who cannot. Once again, I'm not going to get into that. I will save that for Dr. Crows and Dr. Osborne. But when you guys get to countercurrent multiplier, countercurrent exchanger, you will understand that the reason one limb allows certain things to come out, but the other limb doesn't, is because histologically they're different. As we go down the loop of Henle, we are simple squamous. And as we go up the loop of Henle, we're cuboidal. So it's going to change who can move across, okay? So that's where that important difference is. So all you have to remember is everything is simple cuboidal except the descending limb of the loop of Henle. The collecting duct and the sort of terminal end of the distal convoluted tubule have some specialized cells in there, like our principal cells and A and B cells. But once again, that's something you guys will learn when you do your urinary physiology and acid-base balance in physiology, okay? All right, any questions so far? I know we had a really good question about hypertension in the kidney. Do you guys have any other questions? You can go ahead and raise your hand or put them in chat because I can actually see it now. Look at that. Anything so far, guys? I'm hoping that those of you that had lab, or came to lab, I should say, I have to say came to lab because we come at different times now, came to lab this morning or yesterday. I'm hoping some of those structures we went through with the Kahoot are now starting to kind of go, oh, make some little aha moments, okay? All right. So like I said, not going to get into too much physiology here because urinary physiology is incredibly complex. But I do like to give you guys some physiology so you have an idea of what the heck we're learning. When we make urine, it's a 3-step process. Our first step is filtration. Our second step is reabsorption. And our third step is going to be secretion. We've talked a lot about this first step already, right? This is where blood comes into the glomerulus and it's going to get filtered out through high pressure based off of size, not based off of what we want to get rid of. Then we have what's called tubular reabsorption. Tubular reabsorption is where we take things that initially got filtered because they were small enough, and we go, ooh, we want to actually keep that. So that returns solutes back to the bloodstream, okay, reabsorbing it into the bloodstream. It's reabsorption because where did it start? Your circulatory system. So we're going to reabsorb it back in. And then the last one we have is tubular secretion, where things that were too big or ions and things like that weren't initially filtered, but now I want to get rid of them. So I'm going to take those substances out of my bloodstream and secrete them into the tubule. So reabsorption is for things we want to keep. Secretion is for stuff we want to get rid of in our urine, okay? Once again, we don't need to know what particular components those are. That's physiology. But the thing you need to understand is there's sort of a baseline of things in a healthy individual that we're going to reabsorb and secrete. And then it also depends on your diet and your medical conditions, medications you're taking will all affect what's going on here in the kidney. Okay, so you guys will learn more about that later in physiology. So far in the kidney, or I should say in the urinary system, we've learned about the kidney, 3 different layers. We have the pyramids. Within those pyramids, we have those tubules called nephrons that are getting fed by all that vasculature that gets filtered. And then we move stuff around that nephron to inevitably form urine that goes into the collecting duct, the papillary duct, the minor, the major calyx, the pelvis, and then finally our ureters. So the ureters are going to be the little guys that are going to come out of the kidney, 1 for each kidney, typically. And their job is to take that urine to the urinary bladder, which is where it's stored until we excrete it. Now, how we get the urine from the kidney through the ureter to the bladder is through a combination of things like gravity, but kind of like what we saw in the digestive system, our bodies can be in different positions. So if we're laying down and we're only relying on gravity, nothing's going to move. So we also have to use some smooth muscle. So we have peristaltic waves. So peristalsis is, let me write that, peristaltic or peristalsis is the process. It's that rhythmic wave of contraction. So on like the first or second slide of the digestive system, it listed all the things that the digestive system does. One of them being propulsion. If you watch that video, it has really good techno music, and it's of the inside of the stomach where you can see the peristalsis happening, the rhythmic waves of contraction that help push everything through your digestive system. We see those as well in the ureters. That's why if you have a kidney stone that's lodged here in the ureter, or it's making its way through the ureter, you'll feel like these waves of pain, because every time the smooth muscle contracts around it, it creates pain, okay? Now, unlike what we saw in the digestive system, where as we sort of went from one organ to another, there was a sphincter that we had to open and then close to regulate the organ volume. This is just a free-for-all drip into the urinary bladder, okay? As the bladder expands and fills with fluid, the openings here are going to get compressed. So the openings, they actually come in at the bottom. They'll actually close just as the bladder stretches. So that kind of indicates, hey, we're full, let's void, and then they'll open up again and we can continue to drip urine into the bladder. So here's a cross section of a ureter. Kind of looks like the small intestine without the villi. We can see we have a mucosal layer. Remember that transitional epithelium? Remember, this is found lining the ureter and urinary bladder. And remember, its role was to change shape. For stretch. So it allows those organs to expand as they fill with fluid. We have a layer of smooth muscle, okay, that's going to help move things through. And then, oh, look at that, it's adventitia again. Remember, adventitia differed from serosa in the fact that there was no epithelium. It's just connective tissue. So we have adventitia that wraps around the ureters, and we have adventitia around our esophagus. So those are the two places that we've seen it so far. These ureters, the tubes, they're actually going to come in behind the bladder. So they don't come in and stick into the top of the bladder. They come down behind the bladder to the bottom, okay? So that's why we can't see them on this view. So here's one ureter, here's another ureter, and here's their entrances. So they come around from the back. Now these entrances, along with the exit, which is called the internal urethral orifice, form this little triangle at the base of the bladder that we call the trigone. That's where the two entrances and the exit meet. It's on the base of the bladder. Now the bladder itself is important because this is where we're storing our urine. Remember, in the digestive system, we had the liver that made bile and stored it in the gallbladder. And then we have the kidneys that make urine and store it in the urinary bladder. So bladders hold things. As the bladder stretches, it will lead to the micturition reflex, which is very similar to your defecation reflex, okay? And we'll talk more about peeing when we talk about incontinence in a minute, okay? So when we look inside, we can see a few structures, and I just want to point one of them out, or two of them out right now. We'll look at them again in just a second. But if you notice on the inside of the bladder, we have rugae again, okay? And those rugae, once again, just like in the stomach, are going to help with, they're going to go ahead and help with stretch. So the stomach had all these ridges called rugae that lets it stretch, and that's how your bladder can stretch as well. Same thing. The other one I wanted to point out to you is the main muscle of our bladder, and that's the detrusor muscle. It's the big, thick muscle that's going to run around the bladder, okay? So let's take a look at histology, which is perfect because guess what? This is histology for your lab this week. So the mucosal layer, remember, is made of transitional epithelium, and I put a picture of it here in case you forgot what it looks like. It's big, it's fluffy. When you look at the bladder and you look at, like, if you're looking at like a larger section of it, typically when we look at it, we kind of look in here a lot. Or we get just a little bit into that detrusor, it's fluffy. It's like a cloud on that lumen side compared to something like the small intestine, which has these undulations, okay? We have our muscular layer, which is really thick. That's the detrusor muscle. And then, guess what? The bladder has adventitia and serosa. What? How can you have both? Well, here's how. Here's a bladder. The superior portion of the bladder kind of sits sort of right below the abdominal cavity, okay? So this part, oh my gosh, where'd my pen go? This part up here, I'm coloring yellow. This is gonna be serosa, the part that would be like, you know, where your intestines and stuff would sit on top of, okay? There we go. Whereas the back part and the posterior surface, which we'll say this is a posterior view, where the ureters come in. Kind of looks like a weird uterus right now. But the posterior and the inferior parts are all adventitia. It's continuous with the ureters. So the ureters come in from the back, and then they insert on the bottom. So that whole area, even in the front, is going to be adventitia. But the top is in connection with our GI system, okay, like just by touch. So that's going to be serosa. This is a very weird organ. It contains both adventitia and it contains serosa because of its orientation with the digestive system and with its own ureters. That's kind of one of the weird things you need to remember about the bladder in terms of its layering, okay? On the microscope, we're not there where you could tell me it's adventitia or you could tell me it was serosa. So on like your histological drawings, just make sure you annotate it could be adventitia or serosa. That lets me know, hey, they understand that depending on where we're looking in the bladder, it would be one of those two layers, okay? Now, our clinical connection for this unit is incontinence, okay? So incontinence, there's lots of different types. It is basically where you are unable to control normal urination. So let's talk about normal urination first. When we urinate, what happens is it's very similar, like I said, to defecation. The bladder is going to fill with urine. And as it stretches, it sends signals to the brain, I gotta go to the bathroom. And when it does that, we have two muscles, okay? We have over here, next to the internal urethral orifice, that's the internal urethral sphincter. And then we go down the urethra a little bit and then to the pelvic diaphragm, we have the external urethral sphincter. That's an important landmark. So we basically have the internal here, and then we have the external here. So just like with defecation, bladder stretches, internal sphincter relaxes, we get the signal, oh, I gotta go to the bathroom. But in normal individuals with normal muscular tone and physiology, the external urethral sphincter will remain shut until we tell it to relax, and then we can urinate. Now, incontinence means you don't have control of that external sphincter. So incontinence, once again, it can be urinary incontinence, which is what we're talking about now, where you wouldn't be able to hold your urine in. You just kind of go. And then you can have it with fecal matter as well. Okay, it just depends on what the issue is. We see incontinence in a lot of different populations. You can see it in the older population, in elderly patients, because as we get older, things just don't work as well sometimes. We also see it in children, as I was mentioning when we were talking about defecation. Kids, and I should say infants, haven't quite figured out that connection, that neuronal connection between the external sphincter and the brain. So when they have their bladder fill, both muscles relax and they just go to the bathroom, okay? So that's what, once again, potty training does, is it trains that neuronal network, hold on tight to that sphincter until we're ready to go. In adults that are our age, we can also get incontinence, and it's often due to weak pelvic floor muscles like the levator ani. Okay, because the external sphincter runs through the pelvic diaphragm, it's intimately tied to its level of tone. Now, why would the levator ani be loose? Some people, it's just that way. Other people, like those that have had children, when you have a child, those muscles get stretched. And so it can lead to weakness there. As you're going through pregnancy, you're also putting a lot of strain on those muscles. And if they're already weak to begin with, then they'll be weaker during pregnancy. So stress incontinence is where you put stress on the bladder, not necessarily that you have to pee, but it's just there's urine in the bladder, but you cough or you sneeze or you laugh, and that creates a change in abdominal pressure. And as you can see from the diagram, that's going to put pressure on the bladder. Now, normal people, right, if you have a functioning, very well-toned pelvic floor, that's not going to do anything. You have a strong set of muscles, that pressure is not going to matter, everything's going to stay closed. But in women that are pregnant or have been pregnant and had kids, these muscles can get relaxed, and oftentimes women don't seek out treatment to help fix that. So now when they cough, sneeze, or laugh, they pee a little bit because the pressure from the abdomen creating that forceful exhalation pushes urine through towards the urethra, towards the external sphincter, which is now weak, and a little bit of urine comes out. You don't like full on pee your pants. It's just like a little pee comes out, okay? It's a very common thing that happens, but it's also something that can be treated. So doing things like Kegels can help strengthen that pelvic floor. There's even special physical therapy that women can go through to train or retrain and tone those muscles too. So there's treatment. You don't have to live that way forever, okay? All right. We've made the urine. We've got it out of the kidney in our ureters. We've made it to the bladder, and now we have to pee. So now what's going to happen is we're going to need to use our urethra. So here's one of the major problems with the urinary system. Everything starts with U. We have urinary bladder, we have ureter, and you have urethra, okay? So the ureter is to the bladder, the urethra is out, okay? Now, the urethra is actually different depending on what sex you are. So females have a much shorter urethra, which is right here. It's a much shorter urethra than males do. Males' urethras extend all the way from the base of the bladder to the tip of the penis, okay? And one of the other major physiological differences is that with females, ours is not connected to the reproductive system. So here's my bladder, there's my urethra, it's anterior, and then here you can see the uterus and the vagina. So the vagina is posterior, the urethra orifice is anterior. And the uterus, we'll talk about this more when we get to reproductive physiology next week. The uterus sits on top of the bladder, which explains a lot of what happens during pregnancy. In males, however, they share the same exit point with their semen. So the semen is going to come in, albeit from a different angle over here, through the prostate gland and then run through the penile urethra, whereas urine just goes from the bladder through the prostate and then out the penile urethra. That's actually important to know because a lot of the glands we'll talk about and their secretions are going to help try to neutralize the acid that's in urine because sperm don't like that. So that's why we have some of those secretions in semen that we see. So in males, because they do have sort of a shared pathway. What they have are two urethras based off of where it's coming from. I'm going to try to draw this diagram a little bit bigger so you can see it. Okay, there we go. So coming through, we have our bladder here, okay, and then it's going to lead into the prostate. So that's our prostatic urethra. And then it's going to run through the penis. This is called the penile or spongy urethra. More later in chapter 26 on why we would call it spongy. Okay, so males have, it's not that they have two different urethras, it's that they have two sections of it. One that runs through the prostate and one that runs through the penis. And the fact that part of the urethra runs through the prostate is actually really important for male physiology. Some men can get what's called, you don't need to know this, so I'm not even going to write it down, it's really long. Benign prostatic hyperplasia, which is where the prostate grows for non-malignant reasons. And when it does that, it actually can constrict the prostatic urethra, and that will affect the flow men have when they pee. Okay, this also occurs with prostate cancer. So one of the things men in their 40s and 50s and older, their doctors will tell them to pay attention to is, if you notice your stream isn't what it used to be, you need to come see me because there could be something wrong with your prostate. It could be cancer or it could just be benign. My father had prostate cancer, he took it out, he's totally fine. So it's a very, very treatable cancer. But one of the signs is a change in your urination pattern. Okay? Any questions so far about the urethra or urethras, depending on if you have questions for both sexes, before we move on to development? Okay. So the last thing we need to talk about, and then I'm just going to introduce the endocrine system. We won't do a lot of lecture on it, just introduce it, is the development of the urinary system. So the major germ layer that's producing this is mesoderm. Okay. And what's really interesting is during fetal development, we go through 3 pairs of kidneys. The first pair of kidneys is formed and then gone by the 6th week. We call those the pronephros. Nephros is referring to your kidneys. As those guys go away, they basically turn into the mesonephros, which are then once again gone by 2 months. So by the first appointment you have with your OB, you will have already gone through the first 2 sets of kidneys. The final set are the metanephros, and these are the ones that will ultimately form the kidney. Why this happens, I'm not sure. It's probably some old evolutionary thing that we had way back in our history. But a good way to remember these is pronephros, they're primary. First one's formed. Mesonephros has an S in it, so they're secondary. Metanephros has a T in it, so they're tertiary or third. Okay, so pronephros, mesonephros, metanephros. Okay, and then the metanephros basically becomes the kidney that we looked at today. The mesoderm within the metanephros, we call it the metanephric mesoderm. That's where your nephrons come from, which makes sense because your kidneys are made, they're called metanephros, and the nephrons are in the kidney, so it makes sense that they would develop within the tissue in that kidney. So metanephric mesoderm forms the nephrons. Your bladder comes from something called the urogenital sinus. So as your urinary system and your reproductive system are tied very closely together, as we kind of saw in that previous slide. So sometimes it's referred to as the urogenital system, and it's that way sort of sometimes when we're developing. They're very closely connected to one another. So if we look, here's that urogenital sinus right here. Okay, that is what's going to become your bladder. And then here's your 4th week. Here's your pronephros. Those mesonephros are starting to grow. And here they are. And then we get our metanephros at the very end. Yeah, no worries. I'm actually, Stephanie, I'm not even really going to be going into anything you would need to take notes on. I'm just going to introduce what the system is. So we're going to be ending soon anyway. So if you need to print them out, that's okay. But we're just going to be doing an introduction, and I'll be doing more lecture Monday. Okay, so don't feel like you have to rush. Okay, so pronephros, mesonephros, metanephros become our actual kidneys, and the urogenital sinus is what is going to form our bladder. Okay, that is it for the urinary system.